Tiny anti-cancer weapon. Nano-sized particles trigger tumor cell self-destruction. Date: January 16, 2025. Overview. Recent advancements in cancer treatment have seen the development of a groundbreaking method utilizing nanotechnology. Scientists from the University of Pennsylvania have created tiny nanoparticles designed to induce self-destruction in tumor cells. This innovative approach marks a significant step forward in targeted cancer therapies, offering the potential for more effective treatments with reduced side effects. The research team and development process. The research was spearheaded by a multidisciplinary team at the University of Pennsylvania, specializing in biomedical engineering, molecular biology, and nanotechnology. The project commenced in early 2025, with the goal of exploring the application of nanoparticles in cancer therapy. The team conducted extensive laboratory experiments to refine the design and functionality of the nanoparticles. The nanoparticles are engineered at the nanoscale, allowing them to interact with biological systems in novel ways. Their small size enables them to penetrate tumor microenvironments effectively, targeting cancerous cells without affecting surrounding healthy tissue. Mechanism of action. The nanoparticles operate by releasing specific chemical agents once they reach the tumor microenvironment. This release triggers a cascade of biochemical reactions within tumor cells, activating their intrinsic self-destruction mechanisms, also known as apoptosis. This process not only decreases the viability of cancer cells but also minimizes collateral damage to healthy cells, a common issue with traditional chemotherapy approaches. The targeted delivery system is a game-changer, allowing for higher concentrations of therapeutic agents to be delivered directly to the tumor site, thus enhancing treatment efficacy while reducing systemic side effects. Initial findings and implications. Preliminary laboratory results have shown promising outcomes, with the nanoparticles successfully reducing tumor cell proliferation in controlled experiments. The researchers reported a significant decrease in the survival rate of cancer cells exposed to the nanoparticles. The implications of this technology are vast. If successful in clinical trials, this method could revolutionize cancer treatment, providing a more efficient and less harmful alternative to conventional therapies. The ability to target cancer cells directly could also lead to better outcomes in terms of patient quality of life during treatment. Publication and public response. The findings were officially released in January 2025, garnering widespread attention from both the medical community and the public. The University of Pennsylvania's press release highlighted the innovative nature of the research and the potential for future applications in oncology. Several media outlets, including Science Daily, reported on the technological aspects and preliminary data from the experiments. The coverage emphasized the novelty of the nanoparticles and their potential to change the landscape of cancer treatment. Future directions. While the initial results are promising, researchers emphasize the need for further studies, including in vivo experiments and clinical trials, to fully understand the safety and efficacy of the nanoparticles in patients. The research team is actively seeking funding and collaboration for these next steps, aiming to transition from laboratory research to clinical application. Conclusion. The development of these nanoparticles represents a significant advancement in the fight against cancer, showcasing the potential of nanotechnology in medical applications. As research progresses, there is hope that this tiny anti-cancer weapon could lead to more effective and less invasive treatment options for patients battling cancer. For further information, contact the University of Pennsylvania's Media Relations at Induce Tu. or visit their official website hertz.www.penmedicine.org news releases January Penn Medicine scientists develop tiny anti-cancer weapon. The development of tiny anti-cancer weapons using nano-sized particles that trigger tumor cell self-destruction presents several counterintuitive aspects. Here are some of the key points. 1. Cancer treatment through self-destruction. Traditional cancer treatments often focus on directly killing cancer cells or inhibiting their growth. The approach of using nanoparticles to induce self-destruction in tumor cells might seem less aggressive, yet it could be more effective and less harmful to surrounding healthy cells. This counterintuitive method relies on the cancer cell's internal mechanisms rather than external toxicity. 2. Targeted delivery. The nanoparticles are designed to specifically target tumor cells, which contrasts with many conventional therapies that can affect both cancerous and healthy cells indiscriminately. This precision could lead to better outcomes and fewer side effects, challenging the notion that more aggressive treatments are always necessary for effective cancer therapy. 3. Size matters. The use of nano-sized particles might seem like a limitation due to their small size, but this actually enhances their ability to penetrate tumor environments more effectively than larger particles or conventional drugs. This challenges the intuitive belief that larger particles would be more effective in reaching target cells. 4. Mechanism of action. The nanoparticles trigger self-destruction by releasing specific chemicals within the tumor cells. This reliance on the tumor cell's own mechanisms for death could seem risky, as it depends on the cancer cells responding appropriately. However, it represents a sophisticated understanding of cancer biology and suggests that leveraging natural processes may yield better therapeutic results than forcing death through external means. 5. Lower side effects. While many assume that more potent treatments will lead to more severe side effects, the targeted nature of these nanoparticles could lead to fewer adverse reactions. This notion contradicts the common perception that effective cancer therapies must come with significant toxicity. 6. Research and development timeline. The rapid progression from concept to initial results, with experiments showing a reduction in tumor cell proliferation, challenges the expectation that developing new cancer treatments is a slow and arduous process. It highlights the potential for innovation in nanotechnology and cancer research to accelerate treatment development. 7. Public and medical community response. The significant attention and hope surrounding these nanoparticles might be surprising given the historical skepticism regarding new cancer treatments. The buzz indicates a growing acceptance of novel approaches in oncology, contrasting with previous hesitance towards unproven technologies. In summary, the use of nanoparticles to trigger the self-destruction of tumor cells introduces a range of counterintuitive insights about cancer treatment, highlighting a shift in understanding how to effectively target and treat cancer while minimizing damage to healthy tissues.
在最近的研究中，宾夕法尼亚大学的科学家们开发出一种新型的纳米颗粒，这种微型抗癌武器能够触发肿瘤细胞的自我毁灭。这项研究的创新之处在于其对癌症治疗的潜在影响，尤其是在靶向性和副作用减少方面。然而，在这一报告中，有几个重要内容常常被忽视，值得深入探讨。1. 纳米颗粒的靶向性设计。尽管大多数报道强调了纳米颗粒的抗癌效果，但其设计过程中的靶向性常常被低估。研究团队通过精确的设计，使纳米颗粒能够选择性地进入肿瘤微环境。这种特定的靶向性十分重要，因为它直接影响了治疗的有效性和安全性。许多癌症治疗方法，如化疗，往往对健康细胞造成严重伤害，而这种纳米颗粒通过选择性靶向肿瘤细胞， 能够更有效地减少对周围健康组织的损害。这一特性意味着未来有可能开发出更具针对性和更少副作用的治疗方案。2. 自我毁灭机制的机制。报告中提到的肿瘤细胞自我毁灭机制是一个复杂的生物过程。虽然很多人可能会关注纳米颗粒的外部表现，如肿瘤细胞的减少，但忽视了这些颗粒如何激活内在的细胞程序性死亡，如凋亡或自噬。理解这一机制不仅有助于科学家优化纳米颗粒的设计，还能为开发其他靶向治疗提供重要线索。研究细胞自我毁灭的路径和信号通路，可能会揭示更多关于癌症进展和耐药性的知识。3. 实验室条件与临床应用的差异。虽然初步实验结果显示纳米颗粒在实验室条件下有效减少了肿瘤细胞的增殖，但许多人可能会忽视实验室与实际临床应用之间的差异。实验室环境控制良好，能够提供理想的条件，而临床环境则复杂得多，涉及到多种生物和环境因素。因此，研究团队需要进行进一步的临床试验，以验证该技术在真实患者中的有效性和安全性。这一过程可能需要数年时间，而许多人对此缺乏耐心。4. 资金来源与研究背景。宾夕法尼亚大学的研究团队背景和资金来源在某些报道中被提及，但其重要性常常被忽视。研究的资金来源可能影响到其研究的独立性和全面性。了解研究的资助方以及其可能的利益关系，能够帮助公众更好地理解研究结果的可信度。此外，研究团队的背景和专业领域也能够为其研究成果的有效性提供某种程度的保障。科学界需要关注这些因素，以提高对研究结果的解读能力。5. 的纳米技术的伦理和安全问题。在讨论新技术的潜力时，伦理和安全问题常常被忽略。纳米颗粒在体内的行为及其长期影响尚未完全了解。研究小组需要考虑到纳米颗粒的生物相容性、可能的毒性以及其在体内的代谢路径。公众对于新技术的接受程度也可能受到这些因素的影响。因此，在推进临床应用之前，科学家们必须进行全面的评估与研究，以确保患者安全。6. 跨学科合作的重要性。这项研究的成功不仅依赖于纳米技术领域的专家，还涉及化学、生物学、医学等多个学科的合作。跨学科的合作能够促成不同领域知识的融合，推动技术的创新与应用。然而，这种合作在许多报道中未能得到应有的重视。认识到跨学科研究的重要性，有助于推动未来更多类似的创新，并为癌症治疗提供更为全面的解决方案。结论：总的来说，宾夕法尼亚大学科学家开发的纳米颗粒抗癌武器为癌症治疗带来了新的希望，但在这一领域的研究和应用中，还有许多重要内容常常被忽视。靶向性设计、细胞自我毁灭机制、实验室与临床的差异、资金来源与研究背景、伦理与安全问题，以及跨学科合作的重要性，都是未来研究中需要关注的方面。深入理解这些因素，不仅能够为科学家优化纳米颗粒的设计提供依据，也能够为患者的治疗带来更大的益处。
1. Targeted delivery mechanism. The development of nano-sized particles that specifically target tumor cells represents a significant advancement in cancer treatment. The ability of these particles to accumulate in the tumor microenvironment and selectively trigger self-destruction mechanisms within cancer cells minimizes damage to surrounding healthy tissues. This targeted approach could enhance treatment efficacy and reduce the side effects often associated with conventional therapies. 2. Innovative design and functionality. The research highlights the precision engineering of these nanoparticles, which not only allows for rapid accumulation within tumor cells but also facilitates the release of active chemical agents in response to specific environmental cues. This dual functionality, targeting and activation, marks a pivotal step in the evolution of nanomedicine, offering the potential for more personalized and effective cancer therapies. 3. preliminary experimental success. Initial laboratory experiments demonstrating the efficacy of these nanoparticles in reducing tumor cell proliferation provide promising evidence for their potential application in clinical settings. The positive early results underscore the importance of continued research and optimization to translate these findings into viable treatment options for patients. For potential for improved cancer treatment paradigms, the emergence of this technology not only represents a breakthrough in the fight against cancer but also opens new avenues for research in nanotechnology and oncology. With ongoing studies and clinical trials, there is optimism about the future of this approach in revolutionizing cancer treatment, offering hope for improved outcomes and quality of life for patients facing this challenging disease. 1. The development of penicillin. 1928. The discovery of penicillin by Alexander Fleming marked a revolutionary moment in medical history. Fleming observed that the mold Penicillium notatum produced a substance that killed a wide variety of bacteria. This serendipitous finding led to the development of antibiotics, which have saved countless lives and transformed the treatment of bacterial infections. The subsequent mass production of penicillin during World War II enabled effective treatment for soldiers suffering from infected wounds, ultimately paving the way for modern antibiotics. 2. The use of radiation in cancer treatment. 1896. The introduction of radiation therapy as a treatment for cancer began shortly after Wilhelm Conrad Röntgen discovered X-rays in 1895. By 1896, physician Emil Grubb treated a patient with breast cancer using X-rays. The technique evolved over the following decades with advancements in technology and understanding of radiation dosage, leading to its widespread adoption as a crucial method for targeting and destroying cancerous tissues. Radiation therapy remains a fundamental component of cancer treatment, often used in conjunction with surgery and chemotherapy. 3. The Human Genome Project. 1990 to 2003. Launched in 1990, the Human Genome Project was an international research initiative aimed at mapping and understanding all the genes of the human species. Completed in 2003, this monumental project provided a comprehensive blueprint of human DNA informing the fields of genetics and biotechnology. Its findings have significantly advanced cancer research by identifying genetic mutations associated with various types of cancer, leading to targeted therapies and personalized medicine approaches. The project has transformed our understanding of cancer biology and opened new avenues for innovative treatments. The recent development of nano-sized particles by scientists at the University of Pennsylvania marks a significant advancement in cancer therapy with the potential to revolutionize how tumors are treated. This innovative approach, which triggers tumor cell self-destruction, is expected to reshape the landscape of oncology in various ways over the coming years. Future predictions and outlook. 1. Enhanced targeting and efficacy. As research on these nano-sized particles progresses, we can expect further refinements in their targeting capabilities. Future iterations may incorporate advanced imaging technologies that allow for real-time tracking of the particles within the body, ensuring they reach the tumor site more efficiently. This could lead to even higher efficacy rates in destroying malignant cells while preserving healthy tissue. 2. Personalized cancer treatments. The ability to customize nanoparticle formulations based on the specific characteristics of a patient's tumor could pave the way for personalized medicine in oncology. By tailoring these treatments to the genetic and molecular profile of an individual's cancer, healthcare providers could significantly improve treatment outcomes and reduce adverse effects. 3. Combination therapies. The integration of nanoparticle technology with existing cancer therapies such as immunotherapy and chemotherapy is likely to emerge as a prominent trend. By combining these approaches, researchers may enhance the overall effectiveness of treatment regimens, allowing for lower doses of traditional drugs and minimizing side effects. 4. Regulatory approvals and clinical trials. As initial laboratory results show promise, the next steps will involve extensive clinical trials to evaluate the safety and effectiveness of these nanoparticles in human patients. Regulatory bodies like the FDA will play a crucial role in assessing these new therapies. If successful, we could see these treatments approved for clinical use within the next decade. 5. Broader applications beyond cancer. While the current focus is on oncology, the principles behind these nanoparticles could be adapted for other medical applications. Potential uses might include targeted delivery systems for antibiotics, antiviral agents, or treatments for autoimmune diseases, showcasing the versatility of this technology. 6. Cost-effectiveness and accessibility. As the manufacturing processes for nanoparticle therapies are optimized, we may see a reduction in production costs, making these innovative treatments more accessible to a broader patient population. Efforts to ensure equitable access will be crucial in addressing disparities in cancer treatment across different demographics and regions. 7. Public awareness and acceptance. The successful communication of the benefits and safety of nanoparticle therapies will be essential in gaining public trust. Educational campaigns will likely be developed to inform patients and healthcare professionals about this new approach, potentially leading to increased acceptance and demand. 8. Sustainability and environmental considerations. With growing concerns over environmental impact, future research may also focus on ensuring that the materials used in nanoparticles are sustainable and biodegradable. This will be important for minimizing the ecological footprint of these advanced therapies. 9. Collaboration and interdisciplinary research. The complexity of cancer treatment requires collaboration across various fields, including nanotechnology, molecular biology, and pharmacology. We can expect to see increased interdisciplinary partnerships that leverage diverse expertise to accelerate the development of effective therapies. 10. Long-term monitoring and outcomes. As patients begin to receive nanoparticle treatments, long-term studies will be crucial to understand not just the immediate effects but also the prolonged outcomes of these therapies. Monitoring for potential late-onset side effects or resistance mechanisms will be necessary to ensure patient safety. Conclusion: the development of nano-sized particles that trigger tumor cell self-destruction signifies a promising leap forward in cancer treatment. With ongoing research and advancements in the field, the future of oncology looks optimistic, potentially leading to more effective, personalized, and accessible treatment options for patients worldwide. As we move forward, the integration of these innovative therapies into clinical practice could redefine our approach to combating cancer, bringing hope to millions affected by this disease.